Well, I started a long time ago, about 1967, interested in bacteria for my first degree. And I was interested I became interested in bacterial genetics. In those days, it was just mapping genes was important, mapping genes to a chromosome. So I mapped genes involved in germination of bacteria in Bacillus subtilis. And I was intrigued by the fact that some genes formed a set on a chromosome together. So when I moved to California to do further work on bacterial sporulation and germination, I met people at the Scripps Clinic who were interested in HLA. And that piqued my interest in the immune system. And I met Walter Bodmer out there and asked him if I could do a postdoc when he returned to Oxford, or when I returned to Oxford in the UK. And Walter said yes, and I went to do a postdoc, and the project in those days involved cloning genes, because actually this was just in its infancy. And with Walter, he said, well, why don't we clone HLA genes? I knew nothing about cloning and nothing about HLA at the time, but with some help from people, that was at the Cancer Research Institute in London, I managed to clone some HLA class 1 and class 2 genes. And then again, working from my interest in bacteria, I then decided to see if they're part of a complex. And of course, the HLA complex, as we all know now, is a set of genes which are all linked together. And we explored the whole of the MHC complex, and indeed it turned out that there is a cluster of genes in the MHC that link with each other and are related to each other in function. Luckily, we discovered some antigen processing genes interleaved between the HLA class 1 and class 2 genes. And that was an exciting time because we found TAP transporters and proteasome components and taposin-related genes, which were all associated with antigen processing and presentation. So that naturally led on, as people started to get interested in receptors for HLA, to wondering if the KIR genes in the leukocyte receptor complex on chromosome 19 were also part of a complex. And indeed, it turns out that the KIR genes are part of a complex too, a very interesting and complex region of chromosome 19, where the genes have duplicated, and that's one of the reasons why they are clustered, because they've all duplicated next door to each other on chromosome 19. And of course, as a number of groups have found, those genes are in a very complex association with each other on chromosome 19, and they're highly polymorphic. So just like HLA, those sets of genes then are in a cluster, and they're critical in the immune system, and they're all highly polymorphic. So that's how I got involved, and that's really, in a nutshell, about 40 years of my research career. That's a very interesting question. The short answer to that question is that nobody really knows. Of course, there's a lot of work going on in terms of hematopoietic stem cell transplantation and KIR, because of course HLA is critical for stem cell transplantation, and KIR have some effect, although the rules governing which KIR are important in terms of transplantation of stem cells, which ones are important for the graft versus leukemia effect and for the relapse rate, it's really not at all clear. It looks as though something is going on there, but when people have started to look at the rules governing that, they've found that the situation is quite complex. So in different laboratories, depending on which technique you use, whether you deplete T cells and how you treat the samples, the different laboratories are finding different effects. So I think there's a way to go in understanding the KIR in transplantation. But But in terms of solid organ transplants, again, there's some work going on trying to understand if KIR are relevant. And again, it's even less well understood if KIR are important for stem cell transplantation, for solid organ transplantation. Only time will tell. But there are other signs that KIR may be important, for example, in CD8 T cells and what they may be doing. It's also not really known what the role of peptide is in KIR, and so there's a lot to find out still. Given the fact that KIR and HLA are both highly complex, I think we need to understand fully by KIR and HLA typing to a very precise degree, first of all, before we can actually start to map out how they may function in those complex scenarios. Yes, looking to the future, I can think of at least 3 potential applications of KIR. First would be in complications of pregnancy, where Ashley Moffat has shown that the axis between KIR and HLA appears to affect the condition called preeclampsia associated with pregnancy. So judicious choice of KIR could, or manipulation of KIR could actually influence the outcome of pregnancy in terms of mitigating against preeclampsia. It also is ideas that the KIR HLA combinations affect birth weight, although how you would manipulate that situation is not at all clear. A second application would be in cancer, and I'm sure everyone is aware that now T cells are being exploited in terms of attacking tumours. And of course, natural killer cells being killer cells can in fact be exploited also for killing tumour cells. And so it may be possible to manipulate KIR molecules on the surface of the NK cells, or indeed to block certain KIR molecules with antibodies in order to use those NK cells against tumours. A third potential application is in novel forms of vaccination, and this work was started by Salim Kakou, to my knowledge, in Southampton in the UK. Salim has this idea that you could vaccinate using certain peptides to stimulate activating KIR molecules against certain viruses. So it may be possible in the future to use that as a novel form of vaccination against these viruses, or even other organisms such as bacteria. So those are 3 potential applications. Another possibility, I guess, is that the KIR may be used, and certain knowledge of KIR may be important in terms of attacking infectious disorders other than viruses. And I think a lot more needs to be known about KIR types and HLA types and how they interact before we can really get a handle on this situation. Certainly, we've known about ABO blood groups for almost 100 years, I guess. So those clearly are important, as are some other rare blood group types. So obviously it's going to be important to be able to type for all of those rare types. There are other genes that are variable, probably not as much as HLA and KIR. However, it will be in the future possible to pick up some variants that are quite subtle ones that are going to be important for transplantation that we really don't appreciate at the moment. I'm thinking of the LALR molecules, for example, linked to the KIR genes on chromosome 19q those are expressed on more on the myeloid monocyte lineage, and those could well be important in future for transplantation. There are other non-classical class 1 molecules, such as MYC, for example, again, that may be exploited. So I think scattered throughout the human genome, there will be many other receptors with variation in that may well be relevant to understanding stem cell transplantation. At the moment, though, those are, as I say, scattered, and there'll be individual polymorphisms in cytokines and chemokines and other receptors that it will be important to monitor and track. Not only that, in the body of the genes, though, there's some work, certainly in autoimmunity these days, looking at these regions between genes, which many years ago were thought to be junk DNA, but now it turns out those are regulatory regions which are important in regulating the levels of gene expression, many of those regions. And some of them will be relevant to type those in, because the levels of expression in some situations are just as important as the particular allele that's expressed. I think that's probably the easiest question today, because I would advise them to go to any tissue typing lab in the world. Because if you go to a tissue typing lab, these people are generally really experts. They have to be, because in order to type samples, you have to be accurate, you have to be precise. And whenever I've talked to tissue typers, I'm so impressed by the way they actually manage to be extremely careful about their typing. It brings to mind when I first looked at tissue typing using sera or antibodies many years ago. I went to a tissue typing lab and they had all these different alleles of HLA. And we started doing DNA typing, and we thought by DNA typing we'd have a much more precise handle on the individual types. In fact, we did, of course. DNA typing has revolutionized the field. However, what transpired was that the types that these tissue typers had discovered using sera and antibodies were extremely accurate. The DNA just underlined the fact that they've been extremely careful all the way through. So tissue typers, I'd actually encourage anyone to talk to tissue typers. Of course, all tissue typing labs these days use DNA technology, and they're very good at this. The second thing that you could do, though, is go to some of the meetings that are organized for transplantation and typing. I'm thinking, for example, in Europe of the EFI meetings, in the States, the ASHA meetings, and in the UK, our BISHA meetings. Each country seems to have its own society. And these societies are very good at getting together and discussing typing and its relevance to transplantation. So there's a host of different platforms and situations where you can get involved. Yes, of course. I think the KIR are very exciting, of course, because they are at the forefront of understanding transplantation with HLA. And if you look at the history of HLA, it's taken 50 years from its beginnings, probably even longer, before HLA has come to fruition, and it's still highly regarded as a key important molecule in transplantation. KIR are on the similar trajectory. We're starting to learn more and more about KIR, and as we learn, they will become more and more important in transplantation. But there is another one of these key receptors that I'm particularly interested in that has remained mysterious for many years, and that's KIR3DL3. This molecule is highly conserved in different species, and yet we knew nothing about its function. It turns out it's highly polymorphic, and we made a novel antibody against KIR3DL3 many years ago, but we couldn't find any expression. So why have we got this highly polymorphic molecule that's conserved in different species that's not apparently expressed in blood? Does it interact with HLA class 1? It looks as though it doesn't interact with HLA class 1. The question is then, what is its ligand? And recent work from a couple of groups shows that KIR3DL3 interacts with a B7 co-receptor-like molecule, HHLA2. So this opens the door for exploitation potentially in immune cancer therapy. The KIR3DL3, as I mentioned, is not expressed in the circulation, which is why we couldn't find the antibody binding to the molecule in circulation. However, it appears that it's expressed in gamma delta and CD8 T cells, a subset of those in lung and digestive tract. So there's still a lot to learn about this molecule. Having said that, there's still a lot we don't know about the other KIR molecules, but KIR3 is one that's clearly unique.